Dear students, after studying this module, you shall be able to know about the molecular asymmetry, learn about the effect of different spatial arrangements of molecules, and identify enantiomers, diastereomers as well as mesocompounds. We shall also tell you how to calculate the enantiomeric axis and how to evaluate the configuration of various stereoisomers. Now let us begin this topic of molecular asymmetry with its introduction. What exactly is molecular asymmetry? Symmetry, the term, refers to something that is well proportioned. It is an important property that is often correlated with beauty. However, complete symmetry is impossible. The absence of or the violation of symmetry can be termed as asymmetry. Asymmetry has important consequences in different realms. Look at the right and the left foot that we have shown in this figure. They are basically mirror images of one another. But no matter how hard you try, you can't make your left foot shoe fit exactly in your right foot shoe. Unlike a ball and its mirror image, a foot and its mirror image are not at all identical, that you can easily perceive and appreciate through this image. Just as shoes and feet come in mirror image pairs, that is a left and a right, many molecules can exist in two spatial configurations that are non-superimposable mirror images and are known as chiral molecules. Surprisingly, almost all biologically active molecules exhibit this property of chirality. Louis Pasteur pioneered the study of molecular asymmetry. Pasteur became puzzled that tartarates and paratartarates are chemically equivalent compounds but behaved differently towards polarized light. Tartarates rotated the plane of polarized light, whereas paratartarates did not do that. In 1847, he made the surprising observation that crystalline paratartarates consisted of a balanced mixture of left-handed and right-handed configuration crystals. Former type of crystals rotated the plane polarized light to left and the latter one towards the right direction, together cancelling each other's effect and thus producing no optical activity. In contrast, the tartarate crystals showed only one configuration and therefore showed optical activity. Almost 3 decades or 30 years later, two scientists, J. H. Van't Hoff and J. A. Lebel, simultaneously but independently proposed the explanation that carbon has a tetrahedral structure and a molecule having at least one carbon atom attached with 4 different groups is called optically active. It is worth noting, students, that Van't Hoff and Lebel made their proposal at a time when very little was known about the structure of atoms or chemical bonds. It was initially controversial, but soon came to be generally accepted and is now regarded as a well-known fact. Discovery of chirality by Pasteur led to the emergence of a very important and fascinating field of stereochemistry. Stereochemistry deals with the study of spatial arrangement of atoms in a molecule and the behavior of chiral compounds in different chemical environments. Now, after having told you about molecular asymmetry, let us come and study in detail chirality. The term chiral was derived from the Greek word for hand, as they are non-superimposable mirror images of each other. That means two chiral molecules are non-superimposable mirror images of each other. This property of asymmetry is termed as chirality. On the contrary, molecules identical to their mirror images are known as achiral. Most chiral molecules lack symmetry elements like a plane of symmetry, This is a plane that divides the molecule into two halves such as that one half is mirror image of the other. Or a center of symmetry. It is a point from which any atom in the molecule lies diametrically opposite to the identical atom at an equal distance. Chirality is more often observed in molecules having at least one carbon atom which is attached to 4 different groups. To understand the difference between chiral and achiral molecules, let us compare two molecules, 1,1-dichloroethane and 1,1-bromochloroethane. Now these are actually shown to you here in this figure. We can clearly see from the figure that 1,1-dichloroethane has a plane of symmetry passing through CH3-CH bond, whereas no plane of symmetry is present in 1,1-bromochloroethane. To add to this further, to test the superimposability of both 1,1-dichloroethane and 1,1-bromochloroethane with their respective mirror images, we will rotate along the axis passing through the CH bond. The spatial arrangement of mirror image after rotation and 1,1-dichloroethane is identical to the original molecule, but no similar observation can be made in case of 1,1-bromochloroethane. To conclude, 1,1-dichloroethane is achiral, whereas 1,1-bromochloroethane is a chiral molecule. A carbon atom bonded to 4 different groups is termed as a stereogenic center, chiral carbon, or it can be called as an asymmetric carbon. The mirror images of 1,1-bromochloroethane have the same constitution and sequence of bonded atoms. However, they differ in the spatial arrangement of atoms. Thus, they are known as stereoisomers. Stereoisomers, which are non-superimposable mirror images of each other, are termed as enantiomers. It is pretty interesting that enantiomers have identical chemical except when they are interacting with chiral reagents or in a chiral environment, and usually physical properties also like solubility, density, boiling point, etc. But it is also interesting to know that they have a striking difference in their ability to rotate the plane of polarized light as it was first observed by Pasteur, as well as the properties that depend on the spatial arrangement of the atoms. Further, a molecule can have more than one stereocenter. More than 2 stereoisomers are possible for such molecules, and for n number of stereogenic centers present in a particular molecule, there will be a maximum of 2 raised to the power n stereoisomers which are possible for that particular molecule. Now, carbohydrates are a very good example to explain the 2 raised to power n rule, as they have more than one stereocenter. Let us take an example of erythrose. It has 2 stereocenters and thus it will be having 2 raised to the power 2, that is 4 stereoisomers, as shown to you in the figure. These are 2 sets of stereoisomers that are mirror images. They are termed as AB and CD, and therefore they are enantiomers. All other sets of stereoisomers which are not mirror images, that is A and C, A and D, B and C and B and D, are known as diastereomers. Diastereomers are stereoisomers which are not mirror images of each other, and unlike enantiomers, diastereomers show different physical as well as chemical properties. Now, moving on to this figure which I have told you earlier about, you can see the relationship of this particular molecule as enantiomers as well as its stereoisomers which exist as diastereomers, and the relationship of diastereoisomerism between AC and BD as well as C and B and D and A. This is the molecule of erythrose that we are talking about. A molecule with one stereogenic center will always be chiral. However, this is not necessarily the case with the molecule having multiple stereocenters. A molecule with two or more stereocenters which is superimposable on its mirror image is known as mesocompounds or a mesocompound. This is due to the presence of plane of symmetry in the molecule. Thus, they are optically inactive. To understand this concept more clearly, students, let us take a famous example of tartaric acid. Here, you can see that in the figure, A and B molecules are non-superimposable mirror images of tartaric acid and therefore they are enantiomers. However, if you look at structure C and D, they are identical due to the presence of plane of symmetry and therefore is an optical inactive form, that is a meso form of tartaric acid. So, tartaric acid has just 3 stereoisomers, although it has 2 chiral centers. So, it should have 2 raised to power 2, that is 4 stereoisomers, but still it has only 3 stereoisomers because it has a plane of symmetry. For molecules having plane of symmetry, number of mesocompounds can be calculated using this formula. For example, if a molecule has n odd number of stereocenters, the number of mesoforms for that particular molecule will be 2 raised to the power n minus 1 divided by 2. Number of optically active forms will be 2 raised to the power n minus 1, and total number of stereoisomers thus will be 2 raised to the power n minus 1 plus 2 raised to the power n minus 1 divided by 2. Supposingly, a molecule has an even number of stereocenters, then number of mesoforms will be 2 raised to the power n divided by 2 minus 1. Number of optically active forms will be 2 raised to the power n minus 1, which was the same as the molecule having an odd number of stereocenters. Thus, the total number of stereoisomers would thus be 2 raised to the power n minus 1 plus 2 raised to the power n by 2 minus 1. What is optical activity? So, after having appreciated what is asymmetry and chirality, let us now move on to optical activity. Optical activity is an ability of a chiral molecule to rotate the plane of polarized light passing through it in a clockwise or an anti-clockwise direction. Enantiomers will rotate the plane of polarization in exactly equal amounts, that is same magnitude, but in opposite directions. An enantiomer that rotates plane polarized light in a clockwise direction is called dextrorotatory, or it has been represented as a plus sign or a small d. While the enantiomer that rotates the light in anti-clockwise direction is called levorotatory, it is represented by a negative sign or L sign. When both D and L isomers are present in equal amounts, the mixture is said to be a racemic mixture. Achiral compounds and racemic mixture do not show optical activity. Optical activity is measured using a device known as polarimeter. The observed rotation alpha measured in degrees by polarimeter, and it is influenced by several factors like the distance travelled by light through a sample, which is also called the path length L, wavelength lambda of the light passing through the sample, temperature, concentration C of the sample, etc. The standard measurement for rotation for a particular compound is called the specific rotation and is denoted by alpha lambda, lambda as the subscript. It is defined as the angle of rotation of plane polarized light measured at a path length L decimeter and with a concentration of 1 gram per ml. Thus, the rotation or the angle of rotation is basically equal to alpha divided by L into C, where alpha is the specific rotation in degrees, centimeter cube, decimeter inverse and gram inverse. T is the temperature and lambda is the wavelength of plane polarized light. Alpha is measured rotation in degrees and L is the path length in decimeter, C being the concentration of the sample in gram per ml. Now, after having talked about optical activity, one asks the question about optical purity or the enantiomeric excess, or to say how much optically pure a given compound is or a chiral compound is. For sample which is not optically pure, that is containing only one enantiomer, we call that sample optically pure. For all those samples which are having other enantiomers too, but it consists of a mixture of enantiomers, a part of the effect of enantiomer present in major amount is cancelled by the enantiomer present in minor amount. We can easily calculate the ratio of both enantiomers present in such mixtures by using the concept of enantiomeric excess and as well as optical purity. Optical purity is numerically equal to enantiomeric excess for chemically pure samples. From the observed specific rotation that we just told you about and its formula, we can calculate the optical purity as follows. The formula thus for optical purity is specific rotation of mixture divided by specific rotation of a pure enantiomer into 100. The enantiomeric excess or the excess of one enantiomer over the other in a mixture of enantiomer is also called the enantiomeric excess. It can be expressed mathematically as enantiomeric excess being equal to percentage of major enantiomer minus percentage of minor enantiomer. Enantiomeric excess can also be calculated from the concentration of major enantiomer and minor enantiomer. Thus, if we see, then another formula for enantiomeric excess would be concentration of the major enantiomer minus that of the minor enantiomer divided by the total concentration, that is addition of major as well as minor enantiomers concentration into 100. Now let us study about configuration of chiral molecules. Specification of the 4 different groups present on a stereogenic center is termed as configuration. The precise arrangement of substituent at a stereogenic center is known as the absolute configuration of the molecule. However, prior to 1951, when it was not possible to assign absolute configuration, the arrangement of atoms in thousands of optically active compounds was determined relative to one another on the basis of chemical interconversions. This method of specifying configuration is known as relative configuration. Relative configurations, which are D and L systems. In carbohydrate chemistry, configuration is assigned to various monosaccharides with respect to glyceraldehyde as the base molecule, since they can be synthesized from one of the enantiomeric form of glyceraldehyde as shown to you in the figure by a complex series of reactions. If in the Fischer projection of glyceraldehyde, the hydroxyl group is present on the right hand side, it is assigned as capital D configuration. Kindly students, do not confuse this with the rotation of the molecule when a polarized light is passed through it. There we assign it as small d and small l. And if you see, if the hydroxyl group is present on the left hand side, just by looking at the molecule using no polarimeter, it is assigned a capital L configuration. It should be noted, students, that capital D and L configuration is independent of dextro and levo designation, as I earlier told you. So, for monosaccharides, position of hydroxyl group on the chiral carbon farthest from the anomeric carbon will determine its relative configuration. In the figure you can see that enantiomers of glucose are illustrated for you. When hydroxyl group on the chiral carbon in red box, that is which is farthest from the anomeric carbon, is present on the right, we can say that it is assigned as D-glucose. When OH is on the left hand side, it is assigned as L-glucose. This system is not used much these days except for certain classes of compounds like carbohydrates as well as amino acids. This was about the relative configuration. Let us now study about the absolute configuration or the RS system given by three scientists Cahn-Ingold and Prelog, and this system is also known as the CIP system after the initial letters of the names of the scientists who have set this system. Absolute configuration is assigned according to a set of rules devised by three European chemists or scientists, R.S. Cahn, C.K. Ingold and V. Prelog, and is also often called the Cahn-Ingold and Prelog system, popularly known as the CIP system. The rules given by them are known as sequence rules, and they assign the nomenclature of stereoisomers by prefixing the name R from the Latin word rectus meaning right, or S from the Latin word sinister meaning left, depending upon the configuration of the chiral center. The rules for CIP system of stereochemical nomenclature are given here. I will state the rules one by one. First rule, students, is to assign the priorities to the four substituents bonded to a chiral carbon rules. That is, higher is the atomic number of the atom directly attached to the chiral center, higher will be its priority. For example, in this case, if you see this order, iodine has the highest priority, whereas hydrogen has the lowest priority. When dealing with isotopes, higher is the atomic mass of the isotope, higher will be the degree of priority. So you can see here, in case of three isotopes of hydrogen, tritium will be having the highest priority and proton, that is 1H1, will be having the lowest priority. Third rule is that when we cannot decide the priority on the basis of atom which is directly attached to the chiral center, then the groups are prioritized on the basis of the next set of atoms which are attached to the atom directly attached to the chiral center. Next set of atoms are evaluated until the first point of difference is achieved. For example, you can see here in this figure, now the atom which is directly attached to chiral center is carbon here. However, we reach a point where we have the bromine which is the highest priority, then chlorine, then the OH, then the proton. Fourth rule is that multiply bonded atoms in substituents are considered to be singly bonded to an equivalent number of similar atoms. Now you can see how the aldehyde becomes this structure and the cyano becomes this structure. Now the second set of rules is that orient the molecule in such a way that the lowest priority substituent points away from you. Now if we have our eye level there, as you can see in the figure, then lowest priority will be farthest from the eye. In case of Fischer projection formula of a compound, the lowest priority group is always kept in the top or bottom position, that is on the vertical line to find the absolute configuration of a stereo center. If the lowest priority group is not on the vertical line, all we have to do is to exchange any two substituents bonded to chiral center twice, such that the lowest priority group is on a vertical line. It should be noted that a single exchange between any two groups will result definitely in the opposite configuration. Thus, two exchanges are required. Now you can see here we have shown in the figure how the two exchange is done to bring the group with the lowest priority on the vertical line. Third set of rules is trace the direction of curved arrow starting from highest point of priority to second highest priority to the third highest priority. If the direction of the curved arrow is clockwise, then the chiral center has R configuration, and if it is anti-clockwise, then the chiral center has S configuration. Now you can see here we have shown you how the R and S are achieved in according to the CIP rules. Now students, we have in a lot of detail and in a wonderful manner we have explained you the asymmetry, the chirality and how to determine the absolute and the relative configurations. After this, I want to summarize this module. In this module, students, we have taught you that molecules having same constitution and sequence of bonded atoms but differing in the spatial arrangement of atoms are known as stereoisomers. Molecules that are non-superimposable on their mirror images are said to be chiral, and two mirror images are known as enantiomers. Most chiral molecules lack symmetry elements like a plane of symmetry or a center of symmetry. Chirality is more often observed in molecules having at least one carbon atom which is attached to 4 different groups. Stereoisomers which are not mirror images of each other are known as diastereomers. Enantiomers having identical chemical and usual physical properties, whereas the diastereomers show different physical and chemical properties. A molecule with 2 or more stereocenters which is superimposable on its mirror image is optically active and is known as meso compound. The calculation of stereoisomers in different conditions has been clearly specified in this particular module. A detailed discussion also has been done on the calculation of specific rotation and enantiomeric excess, which is also discussed properly. Now students, specification of the 4 different groups present on a stereogenic center is termed as a configuration. Relative configuration is the method of specifying arrangement of atoms in optically active compounds relative to chiral compounds on the basis of chemical interconversions. For example, configuration of various monosaccharides is assigned with respect to glyceraldehyde as a base molecule, since they can be synthesized from one of the enantiomeric form of glyceraldehyde. If in the Fischer projection of glyceraldehyde the OH is present on the right, then it is capital D. If it is present on the left, then it is capital L. Position of the hydroxyl group on the chiral carbon farthest from the anomeric carbon of the monosaccharides will determine its relative configuration. Capital D and capital L configuration is independent of dextro and levo designation, as we have clearly discussed in the module. Absolute configuration is another method of specifying the exact arrangement of atoms on a stereogenic center and can be specified using the CIP system, where the number of stereoisomer is prefixed by R or S. Now students, we have discussed in detail about the molecular asymmetry and its various aspects, and I hope you have found this module very informative.